A-Level Biology 9700, Paper 4, Variant 1, October, November 2024, question number 1 and 2. So we will start with question number 1, and question number 1 is different species of animals have neurons with different characteristics. So what are neurons? Neurons are the unit of nervous system, and the three different types of neurons are available, which takes the messages towards the central nervous system or away from the central nervous system. As for example, three types of neurons are there: sensory neuron, motor neuron, and uh, relay neuron. So here on screen you can see the two types of neurons, which are motor neurons. They takes the messages from the central nervous system towards the effectors. So they are also called as effector neurons. So here you can see the two different types of neurons. Of rat motor neuron has been shown, and the snail motor neuron has been shown. And here you can see the differences and similarities in both of these two types of neurons. First of all, you can see the cell body. Cell body, both of these cell bodies of these neurons have the nuclei, and the dendrites are there, and axons are there, but you can see the difference in the diameter of the snail is 40 micrometer, whereas the diameter of the rat motor axon is the 7 micrometer. So it means the diameter of the snail motor neuron is greater than the rat. So axon, but you can see the difference in the axon as well. That is the difference in the presence of myelin sheath or absent of the myelin sheath. You know, the myelin sheath which is produced by the Schwann cells, and you can see the some areas which are without these areas. These areas which are without, you can see the myelin sheath, and this area is called as nodes of Ranvier, nodes of Ranvier. So, the first question is to label these parts A, B, and C. So first of all, you can see the dendrite. Dendrites are there in both of these, and B, you can see the center of the cell body, they are nuclei, and C are the synaptic node or axon terminals. So these are the three parts which have to be labeled. After labeling, we are moving towards the next question. I have already written the answers because more time is required for, you can see, for explanation. So to explain all these things, uh, I have already written to save time, because in this way the video can be very long. So we can see that the second question is the rat motor neuron has an impulse transmission speed of 50 millisecond. The snail motor neuron has an impulse transmission speed of 8 millisecond. Explain why the rat motor neuron has a faster impulse transmission speed than the snail motor neuron. So I would like to explain this, that the snail motor neuron has an impulse transmission speed greater than the snail motor neuron. For this answer, we will again move towards the figure. And in figure, you can see both of these neurons which they have been shown. So there are some key factors which affect the impulse speed. First of all, you can see the difference in the diameter of both of these neurons, already told you. And the second thing is that the myelination. Myelination, and you can say the myelin sheath is present in the myelin sheath, which is present in rat motor neuron, but it is absent in snail. And the myelin sheath is produced by the Schwann cells. So these are the Schwann cells which produce this myelin sheath. And this myelin sheath insulate the axon and allow for saltatory conduction. Saltatory conduction. What is saltatory conduction? So what is saltatory conduction? Saltatory conduction is actually is the process which nerve impulse or the action potential traveling along myelinated axon. So their movement, instead of moving continuously, you can see they are unable to move continuously. As for example, if some of the points are there where the myelin sheath is not present, already told you, this is called as nodes of Ranvier. Nodes of Ranvier. So nodes of Ranviers are there. So due to the absence of myelin sheath over here at the points which are called as nodes of Ranvier, these nodes are very small gaps. So ion channels are concentrated. So here, what will happen over there? The ion channels are concentrated over here. Now, how this saltatory conduction takes place. So the impulse moves more quickly compared to unmyelination axons because it skips myelinated sections. So in this way, they will move in this way. So it will move in this way. It will skip this myelinated part. So we can say that the transmission will be faster because it will skip the myelinated sections. So they will jump from one point to the other. And secondly, the energy efficiency. I already told you fewer ion exchanges occur, reducing the energy required for repolarization by the sodium potassium pump. So this is the second point. And the nodes of Ranviers are the only points where depolarization and action potential generation occur. So these, you can say the saltatory conduction. The saltatory conduction increases the speed, increases the speed of neural communication, allowing rapid reflexes and efficient nervous system. So in this way, I would like to say there is the difference between both of these types of neurons, and already written the answer. You can see over here. The rat motor neuron has a smaller diameter axon, but axon is myelinated, allowing for saltatory conduction. So these factors enable the action potential to travel more rapidly compared to the snail motor neuron, which is unmyelinated and has a less faster impulse transmission rate. So I would like to add one more point over here, that is saltatory conduction significantly significantly increases the speed of neural communication allowing rapid reflexes and efficient nervous system function. So after this explanation, we are moving towards the next question. And next question is figure 2.1. Figure 1.2, sorry, shows an action potential in a rat neuron, and a figure 1.3 shows an action potential in a snail neuron. So question is, contrast the two action potentials shown in figure 1.2 and figure 1.3. And students, you can see distinctly differences between the rat and snail. You can see on one side they have given us the membrane potential on y-axis, and on x-axis they have given us the time. And you can see the clear-cut differences between these two figures 1.2 and 1.3, the action potentials of both of them. So we have to contrast, contrast both of these. So first of all, the duration, we will discuss the duration of the action potential of the rat. You can say the action potential is much shorter in duration. It occurs within approximately 2 to 3 milliseconds. 2 to 3 milliseconds, you can see over here. This is the 2 or 3 between these points. So this is, or whereas you can see in the snail, the action potential is significantly larger, lasting for about 20 to 25 milliseconds. 20 to 25, you can see this is very short, and you can see, and it takes a larger uh, piece of time. So in this way, it shows a very small, for the very small time, and you can see it takes a more time as comparison to the red. So rate of depolarization and repolarization also affecting, or you can see the depolarization. What is depolarization? When the, which is the rising phase. So this is the rising phase in both of them. In the red, you can see, are very rapid. In red, they are very rapid. You can see very rapid, and the slopes of these phases are this way. They are very steep. They are very steep. It is very steep. So on the other hand, the snail depolarization and repolarization are much slower. You can see they are moving in this way, and then very slowly it is moving, taking more time to complete. So shape of the action potential is also different in them. First of all, we have discussed the duration of action potential. Duration of action potential. And secondly, we have discussed the rate of polarization and repolarize and repolarization. So, and the third one is the shape shape of action potential. So here you can see the shape of the action potential. The action, you can say it is very narrow. So uh, it is very narrow. This is in case of rat, whereas it is wider. It is wider in snail. The action potential has a broader, more round shape, but it is very deep. So next, we can say the hyperpolarization. Hyperpolarization phase, you can see after a repolarization, where the membrane potentially briefly dip below the resting phase. So this is the hyperpolarization. So this place is called as hyperpolarization. So what is happening at this point, you can see the feature in duration is very short, and in this way they both are, it is very sharp tip, and you can see a wider tip. So I would like to repeat the differences again. First of all, you can see the duration of action potential and the rate of depolarization, repolarization, and the shape of the action potential. Now, these differences are primarily due to the types and kinetics of the ion channels present in the neuron membrane. So you can see these typically have high density of fast gated sodium channels for rapid depolarization and fast gated potassium channels for rapid repolarization. So we can say that in rat neurons, high density, high density of fast gated sodium channels and potassium channels. So due to these channels, the, you can say, the polarization and depolarization occur very fastly. And on the other hand, the neurons, snail neurons, have different types of ion channels, including slower gated channels or channels with different kinetics. So the snails have slower gated sodium and potassium channels. So we can say the rat neuron exhibit a rapid transient change in membrane potential. So in this way, we can easily contrast both of these. So I have already written the answer as well. I have explained it, and I have written this answer as well. You can read it, you can understand it. So in figure 2 point, the action potential is much shorter duration. I have already written all these contrasting points in both of them, and I have also written the reason for these contrast. So I think it will be enough for you. If you find any other difficulty, you can mail me, email. I have in my email ID has been given in description. Now it was all about question number 1. Now we are moving on question number 2. The leaves of many plants have stomata that show a regular daily rhythm of opening and closing over a period of 24 hours. Explain why it is important for plants to open and close their stomata in a daily rhythm. Daily rhythm. It means, there is a time, there is a time of the day when these stomatas are open, and there is a time when these stomatas are closed. So there are different reasons for the opening and closing of these stomatas. First of all, stomatas, you know very well, they are the small or tiny pores which are produced between the guard cells, which are present on the lower, primarily on the lower side of the leaves. So they allow for gas exchange. So between the plant's internal tissues and the atmosphere. So they are, they are surrounded by the guard cells, and these guard cells regulate the opening and closing of the stomatas. So why these stomatas are open? What is the importance of the opening of these stomatas? First of all, the first reason, importance for photosynthesis. And what is photosynthesis? Photosynthesis is a process by which the green plants manufacture the food, and as a result of this photosynthesis, oxygen is produced. Not only the food, beside the food, oxygen is produced, and carbon dioxide and water are the, you can say, the raw material for this. And during the process of photosynthesis, we can say the stomatas need to be open. So during the day to allow carbon dioxide to enter the leaf for photosynthesis. So another thing is that the photosynthesis, and second we can see the transpiration. The second is transpiration. So the water vapors uh, also escape through open stomata. The extra water should have to be evaporated. So it is evaporated, it is you can say, by the process which is called as transpiration. So it is essential for transporting water and minerals from the roots to the rest of the water. So it will be easy for the water to move if transpiration is there. So this is the, you can say, the uh, uh, two importance of the opening of the stomatas. The other uh, importance of closing these stomatas. Stomatas should also be closed. They should not always open. So they should always or should also be closed. So water conservation. Why they closed? Conserve water, because if they are open all the time, water will be evaporated by plants. So plants needs to minimize water loss, especially in hot or dry conditions. So closing stomata at night or during periods of water stress reduces transpiration. So we can, in other words, we can say, to reduce transpiration, because this way water will be lost and plants will be dehydrated. So to reduce the transpiration or the water loss. And secondly, we can say that to prevent desiccation. Excessive water loss can lead to wilting and even death of the plant, which cannot be acceptable, to reduce the wilting by dehydration. So in this way, it is necessary to open or closing at some times when water is needed, when the water is not needed. So they should be open or closed. So we can say that this rhythm is very compulsory. So in this way, the plants optimize the photosynthesis in the presence of sunlight. When the light availability is there, stomatas are typically open. During the day, when light is available, photosynthesis will take place at that time. And when the temperature and humidity is there, the stomata's opening and closing can also be influenced by temperature. If there is humidity, the stomatas definitely they will be closed because there should not be already humid outside. So in this way, we can say this circadian rhythm prevents have internal biological clock, you can see, or the rhythm, which helps regulate various processes, including the stomatal movements. So anticipation of daily environmental changes. So in this way, the daily rhythm is a vital adaptation for plants to thrive in their environment. So I have written this answer as well, which I want to explain. So the daily rhythm of stomatal opening and closing is crucial for plants to maximize photosynthesis by taking carbon dioxide uptake during the day and minimizing the water loss by closing stomata at night or during periods of water stress. It also helps to adapt the daily environmental changes and optimize resource utilization. So in this way, the opening and closing of stomata is very important. Now we are moving towards the next question, and next question is figure 2.1 shows the results of an experiment to monitor the rhythm over 3 days and nights for the plant Arabidopsis and Thaliana. The percentage of open stomata is shown each day consisted of 14 hours on white bar light. You can see this white bar shows the presence of light, and the each night the black color will show the night of the 10 hours of darkness. 10 hours of darkness, black bar. So this black bar, 10 hours of darkness. So they have shown over here. Figure 2.1 shows that the percentage of open stomata increases in the first 7 hours of the experiment. First 7 hours of the experiment, the open stomata, as you can see. So here you can see the first 7 hours, they are very, so from here, this 20 out of 24 hours, you can see they are wide open. So describe the sequence of changes that occur in the guard cells that lead to the stomata opening. So let's explain this. That is this figure 2.1, and it shows the general plant physiology. So the sequence of changes in guard cells leading to stomata opening. So first of all, I would like to say the light stimulation. Number one is the light stimulation. So what is happening this process uh, begins, light stimulates the guard cells, you can see over here. So in figure 2.1, the stomata opening increases in the first 7 hours coinciding with the day period. It means this white bar, it shows that what happening over there, that is the light stimulates the guard cells. So here you can see the receptor cells are there. So the guard cells possess um, phototropins. We call them the blue light receptors or the phototropins. Activation of phototropins. These are the receptor cells. So these receptor cells will detect the blue light wavelength. So then uh, proton pumping will be start. So after this activation of blue light, the receptors initiate a proton hydrogen pump. The first step will be the pump which actively transports protons out of the guard cells. So this is the, we are only writing the sequence. So I have written this answer, but I'm only the headlines of that answer. That is the, so this actively transport protons out of the guard cells. So these hydrogen ions or the protons, they will move outside the guard cells. And this creates an electrochemical gradient. You can see when they are outside, more hydrogen ions which have the positive charge on them, they will be outside. So due to which the electrochemical gradient is produced. And then as a result of this electrochemical gradient, the potassium influx will start. That is the influx of potassium ions into the guard cells through specific channels. So after this, there will be the influx of potassium ions. So water potential gradient, this will increase the water potential, uh, potential ion concentration will lower the water potential inside the guard cells, because if they are, it lowers the water potential inside. Because if the concentration of these ions is greater, then the water potential will be lower at that time. So due to the water potential gradient, water will move into the guard cells. Into the guard cells. And due to increase in water potential inside the guard cells, you can see it will increases the target pressure. Increase in target pressure. It will increase the target pressure within the guard cells. So, you know, the guard cells are in this way. They will shape in this way. Here you can see they have thick outer membrane as comparison to the other part. So due to which they will be in this form, the bean-shaped structure. They will adopt this bean-shaped structure because they have thickened walls, and due to this thick wall, it is the, you can say, on the side facing the stomatal pores. So increased target pressure causes the thinner outer walls to bulge outward. So these are the thinner, it will bulge out, whereas the thicker wall will be in this condition. So in this way, the shape will be bean shape, because this thicker wall will resist the expansion. So differential thickening and the resulting in deformation causes the guard cells to move outward, opening the stomata pores. So in this way, the guard cells, they will move in this way, and there will be the opening of this stomata open. So in this way, you can see the stomatas are open, and you can see the graph in figure 2.1 illustrates the outcome of this process. You can see the percentage of open stomata increases during the first 7 hours of the experiment, which corresponds to the light period. So this is the result. So this indicates that the guard cells are actively undergoing the sequence of changes described above, uh, as a result of, in response of light. So all these changes will take place, and this process will allow the gas exchange. And when the gas exchange is there, the photosynthesis will occur during the day. The whole process I have written for you, which I have explained. You can read it. Now we are moving towards the next question. And next question is, the experiment was repeated with a Thaliana plant that were left in darkness from 14 to 96 hours. 14 to 96 hours. You can see the black area shows the night. The results are shown in figure. With reference to figure 2.1, move to figure 2.1. You can see this figure 2.1, in which there is a difference between them. And here you can see the difference between these two pictures. So explain with figure uh, 2.2 shows about the role of genes and the role of the environment in controlling the rhythm of stomatal opening and closing in A. thaliana. So here you can see the role of genes and the role of genes and the environment in controlling stomatal rhythm. So figure 2.1, you can see there is light and dark cycles. Light and dark cycles. So these light and dark cycles show a clear day rhythm of stomatal opening and closing, tightly correlated with the light and dark cycles. I will move back. You can see light. You can see the cycle rhythm. You can see the cyclic rhythm. Light, dark, light, dark, light, dark. So there is a rhythm between the light and dark cycles. But here in this picture, you can see there is no rhythm just like that. In this picture, there is no rhythm just like that. So I would like to say that the um, this observation shows our daily rhythm of stomatal opening, and we can see stomata open during the day, which shows clear with the white bars, and the closed during the night, black bars. It means they are closed at that time. But this demonstrates the strong influence of the environment. Environment means if there is light, the stomatas are open. If there is darkness, the stomatas are closed. So the rhythm is entrained, synchronized by the external light cues. So the continuous, but in figure 2.2, you can see the continuous darkness. The continuous darkness is there in this picture. Even in continuous darkness, a rhythm of stomatal opening and closing is still absorbed, light and dark, you can see. So the compare of figure 2.1, the peaks and troughs are not as sharp, and the rhythm gradually dampens over time. You can see rhythm is not a regular sequence. There is no irregularity as in figure 2.1, irregularity in the rhythm. In irregularity, rhythm is there. You can watch this. When we compare both of these, we can see that uh, it indicates that A. thaliana possesses an endogenous clock or biological rhythm that can maintain the stomatal rhythm even in the absence of external light cues. I would like to write over here that is the A. thaliana has a genetic control on rhythm cycle. Maybe they are irregular. They are irregular, but present. So in this way, we can say that figure 2.1, there is an underlying genetic component that allow the rhythm to persist even the constant darkness. So internal log is likely driven by genes that regulates the expression of protons involved in the stomatal opening and closing mechanism. So both genes and environment play crucial roles in controlling stomatal rhythms in A. thaliana. So the environment acts as a primary cue to it contains the internal log. So genes provide the basis for the internal clock, allowing the rhythm to persist even in the absence of external cues. So both play a role, genes and environment, plus environment. So both of they play a role because if genetically they don't have these cyclic um rhythm, so the cyclic rhythm can be absent. But here you can see there is darkness, but cyclic rhythm is there even in the darkness. Due to the cyclic rhythm in darkness, it means this rhythm was present in the genes. It is the genetic expression. So environment also affects. So environment also play a role, but not to an extent that is the genetic expression is all the way over there. So I would like to read this answer, which I have already written for you. Both genes and environment play crucial roles in controlling stomatal rhythms. In Italiana, the environment acts as a primary cue to entrain the internal clock. Genes provide the basis for the internal clock, allowing the rhythm to persist even in the absence of external cues. Genes provide the basis for internal clock, allowing the rhythm to persist even in the absence of external cues. You can see again, you can compare both of these pictures. That is rhythm. Here you can see a clear cut rhythm, a regular rhythm. That is the light, then first of all the light, and then night, open, close, then open and close, open, close. So this is a regular rhythm. But here you can see there is a irregular rhythm. You can see over there. Only the light is over here, but all these show the time of the darkness. But even in the darkness, the plant is showing the rhythm, that is opening little bit, but these are, you can see, they are very sharp. So this rhythm is there, rhythm is there, but even in the darkness, rhythm is there because this rhythm is due to their genetic expression. It is due to the expression of the genes. Environment is playing a role over it due to you can say environmental factor, that is the at night his tomatoes are closed, but they play a very great role. Only the genetic expression is helpful at this time, we can say that, but both of they play a very vital role in this case. So I would like to say it was all about the question number 1 and 2. Students, if you find any difficulty in any question, you can uh, contact with me. My email ID is given in description. You can contact with me, and please like, share, and subscribe. And thanks for watching.